packaging of DNA. The distance between two adjacent nitrogen base pairs or nucleotides is 3.4 angstroms. That is equal to 10 to the power of minus 9 meters. So, packaging of DNA. DNA is the polymer of many nucleotides. Distance between two adjacent nucleotides 3.4 angstrom. If you convert that into the meters, that is 3.4 into 10 to the power of minus 9 meters. Number of base pairs present in one genome, that is 3.3 into 10 to the power of minus 9 base pairs in human genome. Human cell generally diploid condition. So, in two genomes or in a diploid cell, number of base pairs double to this one. Then, what is the length of that DNA? 6.6 into 10 to the power of 9 into 3.4 into 10 to the power of minus 9 meters. That is equal to 2 meters. That means, in a diploid human cell, 2 meter length DNA present. That 2 meter length DNA that is distributed in 46 chromosomes. But the length of DNA is 2 meters, but the size of nucleus is 10 to the power of minus 6 meters. Then how it is possible this 2 meter long DNA adjusted in this small nucleus? It folded many ways. That is called packaging. This DNA that folded in many ways and adjusted in 46 chromosomes, and those 46 chromosomes are arranged in that smaller nucleus. The measurement or the diameter of that nucleus generally 10 to the power of minus 6 meters. Then how it packages, how it folds, how many ways it folds. Now come to this one. Already we know the diameter of DNA is 20 angstroms or 2 nanometers. So, diameter is 2 nanometers. Now, this DNA that present around nucleosomes so the nucleosome, diameter of nucleosome 10 nanometers. Now, these nucleosomes, 6 nucleosomes that are arranged like a cylinder, and the DNA that present around them, and the 6 nucleosomes arranged like a cylinder called solenoid. Diameter is 30 nanometers and it arranged like this one, chromatin fibers. Again condenses, chromatin fibers. Diameter of this chromatin fiber 300 nanometers and it adjusted in 1 chromatid, that is 700 nanometers, and it is a 700 nanometers, and it is a complete chromosome 400 nanometers in diameter. 1400 nanometers in diameter. Nanometer. So, this 2-meter length of DNA adjusted in the 46 chromosome is the method of the packaging. So, what are the different structures that are formed during the packaging of the DNA in the chromosome? DNA. Diameter 2 nanometers. Nucleosome diameter 10 nanometer. 6 nucleosomes arranged like a cylinder that called solenoid. Solenoid diameter 30 nanometers. Solenoids arranged compactly along with other proteins, non-histone proteins, non-histone chromosomal proteins, non-histone chromosomal proteins. Produce chromatin fiber, (300 nanometers). Now, it produces the chromatid. Diameter is 700 nanometers. 2 chromatids that produce a chromosome. (1400 nanometer diameter). 2-meter length DNA adjusted in human being in 46 chromosomes, and the diameter of each chromosome is generally 1400, 700 nanometer each chromatid, and 2 chromatids is the centromere. So, by this it folds by many ways and condenses, becomes the chromosome. So, during this condensation, this DNA coiled around histone proteins present in nucleosome and non-histone proteins that present in the nucleoplasm. So, consider both histone and non-histone proteins. So, a chromosome consists DNA, histone proteins, non-histone proteins.